Good morning, students. Today we will start chapter number 4, Plant Life, Photosynthesis and Respiration. We all know that organisms are divided into two types on the basis of their mode of nutrition. They can be either heterotrophs or autotrophs. Autotroph means the organisms which can have self-nutrition. The word auto means self and troph means nourishment. So the organisms which can have self-nourishment are called autotrophs, and the plants, as they are able to make food for themselves as well as animals, they are the only autotrophs. The other type of organisms are heterotrophs because they get the nutrition from others. Hetero means different and troph means nutrition. So getting the nourishment from someone else is called heterotroph. All the animals come under the category of heterotrophs. The two important processes which are found in the plants are photosynthesis and respiration. Photosynthesis comprises of two words, photo and synthesis. That means making of anything by the help of light. So the plants prepare their food in the presence of sunlight. So the process is called photosynthesis. Another important process undergone by the plants is respiration, in which burning of the food takes place. So photosynthesis is making of food and respiration is burning of food. We will study about both of these processes in detail in the chapter. Photosynthesis is a process by which green plants use the energy from sunlight to convert carbon dioxide and water into glucose. So we can see the photosynthesis through a chemical equation where carbon dioxide reacts with water in the presence of sunlight and chlorophyll to form glucose, which we call as food, and oxygen gas. In the process of photosynthesis, the green plants absorb water from the soil with the help of their roots. The absorbed water is transported through the roots to the stem and then to the leaves, where the process of photosynthesis takes place. Carbon dioxide from the air enters the leaves through stomata, which are tiny pores on the leaves. When all the substances required for photosynthesis are present in the leaf, a chemical reaction takes place in the presence of sunlight. The energy from sunlight is trapped by the chlorophyll pigments present in the leaf and used to combine carbon dioxide with water to form glucose. In the whole process, the oxygen is released as a byproduct. The glucose formed by the process of photosynthesis is stored in the leaves as starch and used by plants as food. Here's an activity to show that green leaves contain starch. The materials required for it are green leaf, beaker, water, alcohol, tripod stand, test tube and iodine solution. The procedure is as follows. We take a green leaf and boil in a beaker containing water for 5 minutes to soften it. Take the softened leaf in a test tube and pour some alcohol in it. Put the test tube in the beaker containing water and let it heat on the flame for about 2 to 3 minutes. Boiling in alcohol removes the chlorophyll from the leaf. Take out the leaf carefully from the test tube and wash it with water. Put a few drops of iodine solution on the leaf. The leaf turns blue-black in color. This activity shows that the green leaves contain starch. The factors affecting the rate of photosynthesis. Light. Light is a very important factor for photosynthesis. The higher the intensity of light, higher the rate of photosynthesis. When the light is dim, the rate of photosynthesis decreases. Sun is the main source of light energy. However, artificial light is also effective in photosynthesis, but it should be of required intensity. Second factor, carbon dioxide. The rate of photosynthesis increases with the increase in concentration of carbon dioxide up to 1%. Beyond this, the plant will not take in any more carbon dioxide. All the land plants get the carbon dioxide from the air, and the aquatic plants get carbon dioxide from water. Next factor is temperature. Higher the temperature, faster the rate of photosynthesis. The rate of photosynthesis rises to the optimum temperature of 35 degrees Celsius, after which the rate of photosynthesis falls down. It gradually stops above 40 degrees Celsius. So the best range for photosynthesis is 35 to 40 degrees Celsius. Next factor is water. Water is a source of hydrogen atoms in glucose. Both water and carbon dioxide molecules break down using the energy of the sun and combine into sugar molecules containing carbon, hydrogen and oxygen. Plants absorb only 1% of water for performing the process of photosynthesis. The last factor is chlorophyll. It is the green part of the leaves. It traps the energy from the sun, which is used for making food in plants. If we increase the amount of chlorophyll, it does not make excess food, but it probably kills the plant. The chlorophyll mainly absorbs the red and violet light from the sunlight and reflects the green light. That is why the leaves appear green in color. Significance of photosynthesis. The life cannot exist without photosynthesis, as all the animals as well as plant, they depend on plants itself for the food. All the animals directly or indirectly depend on plants for their food. If photosynthesis does not take place, they cannot get food and the life will not survive. Second importance of photosynthesis is that it helps in cleaning of the air by absorbing the carbon dioxide. Carbon dioxide is used in the process of photosynthesis, so this way it helps in cleaning of the air. Next important thing or the importance of photosynthesis is that the living organisms get energy for doing their various life activities from the food prepared by photosynthesis. And the last importance of photosynthesis is that it releases oxygen, which is added in the air, which is an essential gas for sustaining life. What happens to the end products of photosynthesis? The glucose is either immediately used up by the cells or it is stored in the form of starch in the leaves. The oxygen produced diffuses in the atmosphere and is used for the respiration of plants and animals. Here we perform an experiment to show that light is necessary for photosynthesis. The materials required are a potted plant with the broad green leaves, iodine solution, black stencil paper and clips. The procedure for it is we take a potted plant and place it in a dark cupboard for 2 days to remove the starch present in the leaves because photosynthesis will not take place in a dark place. Now we cover the leaf with a strip of black stencil paper, then keep the plant in sunlight. After 5 to 6 hours, pluck the experimental leaf and put 2 to 3 drops of iodine solution on it. The uncovered portion of the leaf turns blue-black in color. It shows that photosynthesis has occurred in that part of the leaf which has received the light. We perform one more experiment to test that the chlorophyll is necessary for photosynthesis. The materials required for it are a potted plant with variegated leaves and iodine solution. The method is as follows. We take a potted plant with variegated leaf. Variegated leaf means partly green and partly non-green. Some parts of it are green and some are non-green. And place it in a dark cupboard for 2 days to de-starch. Now place the plant at least for 5 hours in the sun. Pluck a leaf from the plant and carry out the starch test using iodine solution. We will observe that only the green part of the leaf has turned blue-black. Therefore, it is proved that the green portion of the leaf has chlorophyll which perform the process of photosynthesis. We perform an experiment to demonstrate that oxygen is given out by green plants during the photosynthesis. The materials required for it are a beaker with water, funnel, test tube and an aquatic plant, hydrilla. The procedure for it is as follows. We take a few leafy hydrilla plant and keep them in a funnel. Take a beaker and invert the funnel on the support as shown in the figure. Fill the apparatus with water. Take a test tube full of water and invert it over the funnel. Keep the apparatus so faced in the sunlight for a few hours. After a short time, air bubbles arise from the hydrilla plant and get collected in the test tube. The gas inside the test tube may be tested for oxygen by removing it and then by inserting a lighted incense stick into the test tube. Due to presence of oxygen, the incense stick will glow brightly, proving that oxygen is given out during the process of photosynthesis.